hearing from Safiye and Jan Christian. Safiye is a data science manager at Recursion, and Jan Christian Hutter is a principal computational scientist at Genentech.
Hi, everyone. You can see my screen, right? Okay, I assume so. So yeah, today I and JC will tell you about our work, biological cartography. We first want to thank the organizers for inviting us to speak at this great workshop. And here is our disclaimers from Recursion and Genentech. The views we are about to express are our own and may include forward-looking statements. So what's biological cartography? The continuous scaling of genetic perturbation technologies combined with high-dimensional assays such as microscopy and RNA-seq has enabled genome-scale reverse genetic experiments that go beyond single endpoint measurements. Data sets emerging from these experiments can be combined to construct maps of biology where perturbation readouts are placed in unified relatable embedding spaces. This is to capture known biological relationships and discover new ones. Construction of maps involves many technical choices in both experimental and computational protocols, motivating the design of benchmark procedures by which we can evaluate map quality in a systematic, unbiased manner. In this work, starting with biological perturbations, we first proposed the EFR framework for map building. We then proposed two key classes of benchmarks to assess the quality of a map. We describe univariate benchmarks, evaluating the quality of individual perturbations, and then multivariate benchmarks evaluating how well the map can recover known biological relationships. We demonstrate the application and interpretation of these benchmarks through example maps of single-cell RNA-seq and imaging data. Before we go into the details of our work, here's our team who worked hard to make this happen. This was a great collaboration between Recursion and Genentech researchers, aiming to guide the field in building and benchmarking maps of biology by introducing a framework which can be used in such efforts by others. Let's now look at the EFR framework more closely. The embedding step is the first letter E in this framework, is aimed at creating a vector representation of the experimental screening results. We call the smallest experimental entity that's measured in a map context a perturbation unit. This can be a single cell or a well with hundreds of cells, depending on the data modality. Intermediate layers of neural networks are commonly used to generate embeddings for unstructured data like cell images. Also, linear methods like PCA are common for structured data, such as the transcriptomic profiles. However, nonlinear techniques based on neural networks have also been found to be effective for transcriptomic data. So, next step, filtering. In any experimental process, some perturbation units will not satisfy the predefined quality criteria, and they need to be filtered out. Examples of that include wells with too high or too low pixel intensity in a cellular imaging screen, or perturbation units that are not distinguishable from the controls. And then we need alignment. So this is to correct for batch effects. A batch effect is a systematic effect shared by all observations obtained under similar experimental conditions, such as microscopy acquisition artifacts. These batch effects potentially confound the interpretation of desired biological signal from the readouts. A simple alignment approach is to use control units in each batch to center and scale features in that batch. Another linear method aligning not only the first-order statistics, but also the covariance structures is typical variation normalization, or shortly TVN. Nonlinear methods based on nearest neighbor matching or conditional variational autoencoders have been particularly successful for the alignment of single-cell transcriptomic data. There are typically multiple technical or biological replicates representing each perturbation in any data modality, meaning The same perturbation may be applied to dozens of wells or hundreds of cells. So aggregation of these replicates is critical for a robust final representation of a perturbation. Coordinate-wise mean or median aggregation are commonly used methods here. And the final step in map building is identifying relationships between biological entities, such as gene-gene interactions from protein complexes or signaling pathways. Computing distances between aggregated perturbation representations is commonly used as a proxy for relationships, where smaller distance means a stronger relationship. These learned distances can also be used to visualize the global structure of perturbations through further dimensionality reduction methods such as UMAP. This is a framework, and we can use different methodologies in every step of the IFAR pipeline. And we can perform the steps, those IFAR steps, in a different order or multiple times. So how about our benchmarking pipeline? So what do we do here? We divide our map benchmarks into two types, which we call univariate and multivariate benchmarks. Univariate benchmarks assess the ability of an IFAR pipeline to recover signal on individual perturbations, utilizing the replicates of a perturbation after the alignment step. Multivariate benchmarks down here utilize the relationships between aggregate representations of perturbations to evaluate a map's ability to recover biological relationships. JC?
And I'll go into the details of some of these benchmarks, starting with the univariate benchmarks. So these are meant to assess the reproducibility and robustness of the representations we learned. And we demonstrated two metrics here. The first one is the consistency of the perturbation profiles across replicates. It's quantified by the average cosine similarity between those replicates. The second one is the magnitude of the perturbation effect. How much does the perturbation do compared to a null, as quantified by the energy distance? For both metrics, we can assess the significance by assigning a p-value based on a random set of perturbations as our empirical null. The second type of benchmarks, the multivariate benchmarks, focus on known relationships among perturbations. Now, capturing these is really important because that's the typical use case of our maps of biology, namely discovering novel biologically relevant relationships, either between pairs of genes or between genes and small molecules or other compounds. Whereas in this work, we focus on pairs of genes only, though. There are two main types of data sources we query for these multivariate benchmarks: databases of pairwise relationships and of gene clusters. Pairwise relationships refer to known gene pairs that directly interact in a pairwise manner in a pathway biological process or a protein complex. And gene clusters refer to sets of genes, so potentially more than two, involved in a pathway, biological process, or again in the complex. In both cases, as a benchmarking metric, we put the recall at 10%, which is we give the percent of the annotated gene pairs that we capture within the most extreme 10% of the pairwise relationships when taking 5% from both tails of the overall cosine similarity distribution. And now we did apply these benchmarks and our pipeline to two orthogonal data sets: the recently published genome-wide perturb-seq by Riplogel et al. And the recursion phenomic data. For each of these two maps, we're comparing two pipeline choices. On the left, we show those for the transcriptomic map. The first option is a slight variation on the one that was used in the Riplogel et al. Paper, and we call it the PCA pipeline here. The gist is that it employs a linear dimensionality reduction and batch correction techniques, while the second one, which we call SCVI pipeline here, uses nonlinear dimensionality reduction and batch correction as provided by a conditional variational autoencoder.
So for the phenomic data from recursion, we compare two pipelines with different alignment strategies. The center scale pipeline are simpler alignment approach, centers embeddings per batch at 0 and scales to unit variance based on the controls in the batch. The second one, titled TVN here, based on typical variation normalization, decreases the impact of the dimensions that capture variance among the controls.
So depending on the choices that we make, that we just introduced, right, in the EFR pipeline, we can get different levels of recapitulation of known biology, which is the whole point of our benchmarking pipeline. So here are univariate and multivariate benchmark metrics for the 4 maps we just discussed. We report the results for all genes. We skip the filtering step of filtering for perturbations with a significant effect, just so we could have an apples-to-apples comparison using the same set of genes between the pipelines. For pairwise relationships, we consider 2 publicly available sources, reactome protein-protein interactions from complexes with at most 4 proteins, and sigma-sigma pathway interactions. And for the cluster identification metrics, we use reactome gene sets from MSIG to BC2, signal pathways, and quorum protein complexes. So zooming in on the results for the transcriptomic data on the left, we see that there's a slight advantage of using the nonlinear dimensionality reduction batch correction provided by SCVI.
Yeah, and on the right, again, we see the results from recursion phenomic data. We report here we report recall results relative to a baseline convolutional neural network trained to be partially resilient to batch effects. We see that here in this table on the right, an alignment step by TVN or center scale leads to a considerable increase in a majority of the metrics compared to our baseline, and TVN has a clear advantage over center scale.
So in the previous slide, we looked at those benchmarking results globally across thousands of pathways. To provide now a more granular perspective, let's look at how the 2 transcriptomic maps compare in terms of the recall value per cluster when looking at the quorum data set. And that's what we show here in the scatter plot. So consistent with the summary metrics, the SCVI pipeline performs better for most of the clusters, which we can see by the fact that more points fall above the diagonal than below. And then for some selected complexes, we zoomed in and now here show the cosine similarity between all pairs of genes in those clusters in a split heat map on the right. Above the diagonal are the relationships from the PCA pipeline, and below the relationships or distances with the SCVI pipeline. So it looks very similar, but if we zoom in, for example, on the BRG1 associated complex in dark green, we do see that that it exhibits a much clearer clustering structure when we use the SCVI pipeline, as suggested by the recall metrics for that complex that are shown on the left in the scatter plot.
Okay, so as another example, and maybe more zoomed in example of the known and novel biology identified by the benchmarked EFR pipelines here, we examined the cosine similarity structure for the integrator complex, which was also explored in the genome-wide perturb-seq paper that we used data from here. We look at 2 split heat maps, one for the transcriptomic maps we built and one for the phenomic maps. And on the left heat map above the diagonal are relationships from the PCA pipeline for transcriptomic map, and below the diagonal are the ones from the SCVI pipeline. And on the right heat map above the diagonal are relationships from the phenomic center scale pipeline, and below the diagonal are the ones from the TVM pipeline. So known biological structure of the integrator complex is presented on the right so that we can compare. So the thing here is we see that both data types and all 4 maps that we built accurately identify the modular structure of the integrator complex. And the interesting thing is all can identify a previously unknown gene, C7orf26, as part of the integrator complex. This gene was officially renamed in 2015, earlier this year, after 2 papers, including the genome-wide perturb-seq paper, proposed it as part of the integrator complex. This is really a good example of how we can use those maps to discover unknown biology. Okay, let's end with our contributions here, a summary of them. In this work, we describe a framework for systematically constructing whole genome maps of biology and benchmarking their performance globally. As a demonstration, we present several map options built using 2 orthogonal data types, single-cell transcriptomic data where perturbations are based on CRISPR interference, and array-based phenomic screening with CRISPR knockout from recursion. Results demonstrate the impact of different processing pipelines and metric choices. This framework can be used for any large-scale biological map building and benchmarking effort, regardless of data types. It also can be expanded to include settings where additional perturbation types or assay variables are assessed. We hope that this framework will be generally useful as these large-scale map building efforts become more common. And note that both Recursion and Genentech are hiring. Please take a look at our careers pages. And thank you for your attention.
Thank you so much, Safiye and Jan Christian. So we had one question that's come in the chat. Everyone should feel free. We've got 2 minutes till the poster session, so feel free to send in some more. The first question is, how do you build your atlas of known biology that you use in the multivariate benchmark?
Yeah, so I assume the question is the source of the known biology, right? JC, do you agree?
Yeah, that's how I would understand as well.
Maybe
so
you can
we showed some of the sources, right? So any kind of known PPI interactions, for example, is one example. The one that's probably the most convincing are these protein complexes by Quorum, and the one where we also get kind of the best recall metrics for, among others. One could generally think about any kind of knowledge graphs, right? That one could use to build these.
Mm-hmm. And yeah, our poster session is at 2 p.m. Mountain Time, 4 p.m. Eastern. So please come and we can chat.
Okay, great. Thank you so much. Thank you to all of our presenters in this session. Keynotes, recorded talks, contributed talks, they're all great. Thank you so much. And so now there will be an hour-long poster session over on GatherTown. If you have any questions, finding your way there or about anything else, you can find us on Slack. All that is linked from our website, lmrl.org. And so we look forward to great discussions throughout the day.